Hello everyone, let's proceed on to the chapter 3, Basics of Anthropology. The name of the chapter is Genetics and Evolution and the book is Amber and Ember. So let's start with messenger RNA. Messenger RNA, mRNA, is a type of ribonucleic acid that is used in the cell to copy the DNA code for protein synthesis. So protein synthesis is making of proteins from amino acids. So DNA is a code that store the genetic information, but DNA does not directly affect the formation of cells. So cell formation, it is done via a process in which mRNA and tRNA both plays a vital role. So what are these? So one of the ribonucleic acid RNA, which is messenger RNA, it is copied from a portion of DNA and then it moves outside the cell nucleus to direct the formation of proteins. And proteins, as you know, they play a vital role. They have so many functions, and most of our body, most of our organs and tissues, and both internal and external organs, all of them, they are run by proteins. So, and they also act as a catalyst for synthesis of DNA and RNA for the activities of cell. And they also contribute many structural elements that determine the shape and movements of cell. So this process, protein synthesis, it is catalyzed by messenger RNA or initiated and done by messenger RNA, which is like DNA in that it has a linear sequence of bases attached to sugar phosphate backbone. So the phosphodiester backbone bases, it is there, the strands are there, and just that the one of the base uracil, it replaces another base thymine which is present in DNA. So in case of RNA, adenine, it binds with uracil, then CNG, it the whole chain in it goes on like that. And it is also single-stranded rather than double-stranded structure of DNA. mRNA, it is formed when a double-stranded DNA make a molecule. It unwinds and forms a template for mRNA. And when the DNA is copied, mRNA, it is released from DNA, it leaves the nucleus, and the double helix of DNA is formed again, or the uh, double-stranded structure is formed again. So this one is the diagrammatic representation of mRNA. So here it is single-stranded rather than double-strand structure, double helix structure of DNA, which is in which is like a ladder, but the template for mRNA, it is single-stranded. So the other strand, it is not there. And how does it happen? When DNA it unwinds, and the double helical structure, when it is straightened up, here in this case, when this double helical structure, when it is straightened up, it looks like a ladder. Then this is the more you can say unbound representation of the phosphodiester bonds or phosphate sugar backbone, and these are the bases. This is how DNA can be represented in two-dimensional structure. But when the protein synthesis process sets in, here, out of this whole double-stranded structure, one strand is removed, and in that strand, Here, thymine, it is replaced by uracil. So this structure, it will be here like, let's have a clear explanation. So let's say this is the double helical structure of DNA, and when it is unwound, it is formed like a ladder in which the handles, these are phosphodiced bonds, or in simple, these are sugar phosphate bonds. And the pedestal of ladder, they are the nitrogenous bases, which are ATCG in case of DNA. Now when we further unfold it, so this structure, it will be like this. A will bind with T, and the sugar bonds and phosphate bonds will be there, and this process, this whole structure, it will be unwound in this way. Again, phosphate groups. So this sugar phosphate bonding, this is the backbone upon which the bases, the nitrogenous bases, these rests. And when this representation in two-dimensional structure, it is represented this way. I'm trying to make it as simplified as possible. So this is how the whole DNA structure is formed and unwound, especially in two-dimensional structure. And so here, in this chain of A, T, C, G, T, A, G, C, this chain, it goes on, and in this chain there lies the genetic information for life, genetic information for all the processes, all the structure, the whole skeleton, and the structure that rests on that skeleton. But when the the process starts, DNA does not by itself accomplishes all the processes. So how does it does that? How does it initiate or help in all the skeletal and the main body processes? All such processes, they are done in the form of protein synthesis. Since all the cellular and extracellular activities. So cellular and extracellular activity, it means all the activities um, that are expressed within a cell, which includes nucleus, cytoplasm, and all the organelles. Then all the extracellular activities when all such cells they form to express in form of tissues, organs, all such amino acids and proteins which are necessary. So both the processes, they are done by protein synthesis, and protein synthesis is catalyzed by mRNA, and mRNA is itself a template, a cutout section of DNA. So when protein synthesis is about to start, DNA, it severs one section, one template uh, from one of its parts. So this template, it will, when cut out, it will be seen like that. The sugar phosphate bond, it will remain intact. Then the attachment of bases. So A will be there, adenine, cytosine, guanine, and then thymine, it will be replaced by uracil. So the structure of mRNA, the template, just a template, it will be in this form, and it will also be single stranded as compared to the double stranded structure of DNA itself. Now this single strand mRNA, messenger RNA, which act just as a messenger, and um, its chief function is to initiate protein synthesis, and second, it carries the information from DNA to mRNA to outside the nucleus. This is the function that is done by mRNA. Then protein synthesis, which is once the mRNA, it is released from the DNA and travels out of the cell nucleus and into the body of cell. And if you are not aware, if this is a particular cell, in cell there is a nucleus, and outside the nucleus there are vacuoles. We can classify the extracellular material as organelles, mitochondria, plasma, and cytoplasm. So from nucleus, mRNA travels outside the nucleus and into the extracellular matrix. And here it attaches to a soft structure in the cell called ribosome. Yes, so ribosomes are there, which uh, which helps in making protein synthesis, which helps in making protein. So mRNA, it attaches with ribosome, and ribosome, it copies the code that is being carried by mRNA, and then ribosome, it it with the help of the code which is carried by mRNA, makes chains of protein in the form of amino acids and peptide bonds. So here ribosome, it reads the chemical basis, or it reads the code that is being carried by mRNA, and then it commands the ribosome the specific amino acids to join together to form a protein. So here the structure is like that. This is the single strand mRNA that has been cut out of DNA. This template, when it attaches with ribosome, here ribosome reads the information, reads the code, and then directs the production of amino acids. And those amino acids, when they join, they form the protein. So, and here, when there are more than 10 peptide bonds, it is known as polypeptide. And when there are more than 100 peptide bonds, it is known as a protein. So, and peptide bonds, the basic elements, basic unit of a protein. So, protein exists in a form of a chain, just like the polymers. So, this chain itself, It decides which section of polypeptide or how many sections of polypeptide bonds they are attached or segregated to form a protein. And we will talk about it later in the chapter of protein synthesis. So here the mRNA copy of cellular DNA, it is read by ribosome. Then ribosome attaches the amino acid with corresponding transfer RNA to a chain of amino acids. The chain of amino acid also called polypeptide chain because amino acids are linked together by peptide bonds. Then a chain of more than 100 amino acid long, it is known as a protein. So it's like a chain structure, and it depends on the number of nodes or the number of peptide bonds that are read together, and they are segregated and assembled together to form a protein. So here in the case, if amino acid chains are more than 10 structure or 10 bond long, it is known as polypeptide, and 10 such polypeptide chain, they are known as protein, which in itself means more than 100 polypeptide bonds, 100 such nodes when together they will be called a protein. So all in all, it is about the joining of the chain of amino acids. Then there are also mRNA commands that tell the ribosome when to begin, when to stop constructing a protein. Then, thus, the DNA code copied onto mRNA provide all the information that are necessary for ribosome to build the proteins that make up the structure of organism and drive the processes of life. So all in all, let's rewind it. DNA structure, when unwound, it it can be represented by sugar phosphate bonds and nitrogenous bases. The sugar phosphate bond, they forms the backbone upon which the nitrogenous bonds can stay intact even during stresses and the environmental forces known as mutations or UV radiation or any such lethal factor which can alter or destroy a DNA. So this structure, these are the scaffold, is airtight strong structure that provide the necessary resilience to the nitrogenous bases, and the chaining of nitrogenous bases, which is which if unwound and stretch into a long wire or a long rope, it will be simply nothing but a repetition of these nitrogenous bases, which will be read as ATCG, then TAGC. So this structure keep altering itself because the structure itself is double stranded, and any organism, in order to survive, it needs protein in both in inside the cell and outside the cell. So both cellular and extracellular processes, they need protein to function properly. And the protein synthesis, the code of which it is carried in this DNA code, in the DNA. So how does DNA direct the structure of making of protein? Because DNA is contained in nucleus, and the protein making body, it is outside the cell nucleus. So the mechanism by which DNA synthesize protein is via mRNA. So To direct mRNA, DNA, it severs its one such template, and it's one of the base T, it is replaced by uracil, and then this template, which will look like this section, it this section, it will be known as mRNA template. This template, it will be carried out of the cell nucleus into the body of cell, inside the cell, and here in the body there is an organelle called ribosome, and ribosome are the protein makers of cell. So here, when mRNA attaches with the ribosome, ribosome reads the that is instruction that is sent by DNA, and it directs amino acids to be formed in particular chain, and this chain, which is in form of nodes, also known as peptide bonds, and when this chain is more than 10 nodes long, it is known as polypeptide chain, and when this chain is more than 100 bonds long, it is known as a protein. So when such 100 nodes are formed, a protein is formed, which is directed by mRNA. This is the basic of protein synthesis. Now let's talk about sources of variability. So natural selection, it proceeds only when individuals within a population, they vary. This is the first condition of natural selection to take place. If there are no variations, how will natural selection will select anybody? Selection will happen only when if there are different versions to be selected against. So the sources of this new variation, they are genetic recombination and mutation, and there are also two processes which are gene flow and genetic drift, that are also associated with onset of or the integration of variability or variations within a population. Let's first see what a genetic recombination is. Genetic recombination is the distribution of trait from parents to children. They are they varies because no offspring they look alike their parents, and even brothers and sisters, they do not look alike exactly. So how does this happen? So this assortment of genes in sex cells, it provides the conditions for genetic recombination. So genetic recombination in short means that when an offspring is reproduced by its parents, it it copies the genetic code from its parents. So here two processes are there for cell division by which this code can be copied. The first one is mitosis, which deals with all the non-gamete cells. So here the mechanism is simple. One cell, it divides into two, and so the basic code of the first parent cell, it is copied exactly into the daughter cells. No such variation is there. It is a simple exercise. But in case of meiosis, here the copying mechanism, it varies slightly, and this variation is in form that here the offspring, it inherit and an assortment, a random assortment of DNA material from its parents. So Here, this copying mechanism, it is basic in its basic form. It is random. So let's see how random it is. First, the DNA or the DNA in form of chromosomes that are received by parents, it is a matter of chance. That's why it is known as random assortment. So in mitosis, the material that is inherited by daughter cells, this is a parent cell and these are daughter cells. So the genetic material that is being inherited by both the daughter cells, it is similar to the one that is carried by its parent cell. And this mechanism is quite simple. One cell, it divides into two. So there is one nucleus here. All the material, both cellular and extracellular, all are intact. And this process, mitosis, it severs the cell into two parts, and both the cells, both the half cells, they are they will be in this way on the intermediate session, and then both of them, they will be copied in exactly same mechanism. So this is some kind of exponential growth which happens, which keep dividing cells, and such division is necessary for continuation of any individual, because cells, they have a very limited time, and division or death of cells and creation of new cell, it is necessary. So that's how when new cells are created, the parent cells, they are destroyed by the process known as apoptosis, and even the cellular cytosis. But then this situation is different in case of gamete, because first, uh, the derivation of cells in gamete, first of all, it will lead to creation of a new individual. And then second, here, the inheritance itself, it is about inheritance of eggs and sperms. So here, the linear cell division, it should not happen, and it could not happen, because in that case, every individual will have full copy of both eggs and sperm, and the variation or the assortment of individual into one particular gender, it will not happen. And in that case, the gametes themselves will die. So the appropriate procedure, it is that these cells, they need to be derived, they need to be inherited by a different process known as meiosis. And here, a cell that is derived from both the parents, mother and father, here the derivation or inheritance of all the traits or the chromosome, it happened in this way, that the alleles, which is every individual, it carries genes in two forms, both of which they have inherited from their own parents. So here, the derivation of chromosomes or derivation of gene, inheritance of gene in form of these alleles. Now we have four such alleles. If this cell is to inherit two alleles, a form of gene, so if it is to inherit two alleles out of four, here, which allele it will derive, it is a matter of random assortment. So here, natural selection is in full force, and here the derivation of a particular trait or particular chromosome, this is done by assortment. So natural selection and the inherent DNA mechanism, it may select 1 and 3 or 1 and 4, 2 and 3, 2 and 4. So out of these four possibilities, any couple or any two allele, they can be inherited. So this mechanism, it helps in inheritance of best characteristic into the gametes. Best characteristic, and you can say here the gamete cells, they while division or while inheritance, they get to choose between four such possibilities, and I'm talking about only one such characteristic or one chromosome, and we have 23 pairs of chromosomes. So 23 by 4, these many possibilities are there, and which gene will be inherited, which form of allele will be inherited, it it is all a matter of chance. So that's why this process is quite different. So here, each reproductive cell, which means gamete, it carries a random assortment of chromosomes. So this is the random assortment of chromosome, and then this structure, this possibility, which may be around 92. So I have just cited a number 92, that is the minimum number of possibility, and this possibility, this number, it can increase exponentially if there are many such similar alleles. As we know, one single trait, it is regulated by and controlled by the mutual functioning or the cumulative functions of many genes. So any trait with which is controlled by many genes means many alleles. So many tools. Let me elaborate this in a different way. What happens in gamete cell or reproductive cell, it is the first mechanism of natural selection in the form of genetic recombination that is seen in every offspring, and it is seen in all the offspring that are reproduced or that are born in the world. And since this is different from mitosis, the reason for which is that if mitosis goes wrong, it will only create multiple of cells, which can be in form of cancer if this process is not controlled. And then the other mechanism is also there, which is known as cell death. So here, as cell division goes on in exponential number, because here one cell is giving two daughter cells and two daughter cells are there giving four, and this number, it will keep increasing in exponential way. But it doesn't happen that such exponential growth will lead to development of more organs or more cellular structure. That does not happen, because along with cell division, cell death also occurs simultaneously. So all cells which are not necessary, they are killed in a programmed manner, which may take many forms such as apoptosis, cytosis, autophagy, necrosis. So in short, you can call it programmed cell death, which is an inherent function of any individual, which helps in healthy survival of and healthy division of cells. And this mitosis process, it keeps going on this way. And since the inheritance, it is very simple, but in the gametes, the situation is different, because at one, any individual is born just once. So the assortment of genes or assortment of chromosomes that is carried by any individual, it should be a careful exercise. And human, they inherit 23 pairs of chromosomes. So 23 pairs of chromosomes. Chromosomes are nothing but biological structures which can be seen, which can be felt, whereas genes, they are the traits which are not seen but which has uh, these uh, these are characteristics or features of a particular species, a particular individual, that are given to the particular chain of DNA. So this repetitive unit of DNA, let's say the DNA is in form of a long chain which keep repeating itself, and this repetition is very long and very long without any variations, just that ATCG. Then on the other side it will be replaced by TA and GC, and this repetition will goes on. So how does this form of repetitive DNA, this form of chain, is manifesting into particular traits? So it may happen that if you give these number for simplification, like this chain, it started from somewhere, you started a template numbering 1, then the whole chain, it can be named as such. Now let's say from 1 to 10, this structure, it is giving trait for eyes, and the next 12 to 35, this this strand of DNA or this chain of DNA, it is for voice. So here, this strand which together which manifest gene, it is known as manifest eyes, it is known as gene. Same way, the other strand which from 12 to 35, or whatever the number is, this long number, it expresses itself in the form of voice. So this is the gene for voice. So these are the abstract or the ideatic structure which express the form of traits. And DNA, it is wound in the form of chromosomes. These are different structure. These are biological structure, and these do by themselves do not express or do not manifest in anything. But whatever we are talking about, the whole idea of evolution and genetics, it rests on genes. And if you unwind DNA, it will only be a long chain of repetitive units. And even if you want to see it, no other structure, no other pattern will be visible. All you will be seeing is a repetition of ATCG, TAGC, ATCG, TAGC, which by itself means nothing. It means only when a particular strand, whichever its number is, it may be from first strand to 100. So this section of DNA chain, it is characterizing by a particular trait such as teeth. So this whole section, it is known as a gene. And then every individual that you see, even if your mother and father, they themselves have inherited the copies from their own parents, F1, M2, F2. So every individual, he or she, so your mother, she carries two copies of every trait or two copies of every gene which are inherited from her parents. So for teeth, one copy which she has inherited, it is from her mother, and the second which she has inherited is from her father. So that's why in her DNA also, the gene for teeth, it will be expressed, it will be seen, it will exist in form of a double. And these double codes or the double traits or genes, these are known as alleles. These are nothing but two forms of a particular trait. So here you can see the first one, which your mother derived from her own mother, it is a dominant trait. And then the other trait, which she derived from her father, is a recessive trait. And she carries the copies of both the traits. So both will exist, and this existence of both the traits together, it is known as alleles. But together, they are specifying one gene only. So this is the basic structure. Now let's come again to gametes. So gametes, here the situation is different because individual is born just once. So this process, it needs to be uh, carefully monitored, carefully executed process. And our DNA, our cells, they are naturally adopted this way, that they inherit a good number of really naturally selected and the better assorted genes. But one thing is clear, they can assort, or they can inherit what is already inherited by both your parents. So it's not that they can inherit one particular trait which is not our which does not exist in one of your parents. So here your mother, she will carries a lot of genes, a lot of variations for a gene. Let's say it is for teeth, it is for voice, it is for height, it is for skin, and the same corresponding in your father: skin, height, voice, then teeth. Now they themselves have their traits in the form of tools, in form of alleles. So here the traits, they will manifest in this way: skin, both recessive, height, one dominant, one recessive, then voice, both dominant, and teeth, both recessive. And now the offspring, he has the choices for skin, for voice, for height, and for teeth. Now the inheritance of all the characteristics in gametes, which is done by meiosis. So here it will, the possibilities are many. First, I may have expressed this characterization or inheritance or even existence of genes in the form of alleles, and I have only mentioned here two letters. But as I already said, any particular trait, it is controlled by many genes, or mm, or many sections of DNA. It may be that a particular gene, it is controlled by, it is functioned by the chain which is 1 to 100, then 112 to 118, then 212 to 320, this way. So there are three different genes, and all of them, they may have their two forms. So here the possibility of inheritance, or um, the probability of recombination, it increases manifold, which the reason for which is, if you have studied in, if you have solved the probability in mathematics, so this recombination, probability, permutation, combination, all such methods, they are about deriving a particular set from multiple inputs. Inputs are multiple, and you need to derive one copy. So here, if these are 5 marbles, A, B, C, D, and E, and you need to derive one copy or one marble that can be copied in the gamete for the future offspring. So here, which one will be selected? It depends on um, how long or how many the input variables are. If the inputs are so many, the permutation and combination, it will be a tricky process. And if there are only two variants, then the probability, it comes to 1.5, and any such copy can be carried. But in the case of genes and traits, and I have already told you many characteristics, many traits, even for eyes, even for your nose, your voice, your height, your length, your internal structure, all such, they are functioned and regulated by many genes together. And here, meiosis, it becomes a tricky process, because your cell, the process, it needs to select one copy, which is one form of copy, from all the inputs and all the variables possible. So this one here, skin one, let's make it simple. So options here are, in case of the mother, there is one dominant and one recessive. In case of father, both are recessive. So here, the meiosis, it will ensure that one dominant is inherited, and then all the three, they are recessive one. So the assortment, it will happen in this way. Even from the three recessives available, meiosis, it will select the one that is capable of inheriting, that is fit for inheritance. In the same way, we take the inputs for voice. So in case of mother, one is dominant, one is recessive, and in case of father, both are dominant. So here, one dominant out of father will be taken, and now out of the three, in which one there is dominant, other one is recessive, and the other recessive. So the mechanism meiosis, it will inherit the other dominant. So this will be the structure of allele that is inherited in the offspring. In the same way, in case of height, mother carries two dominant variants, and father, he carries one dominant and one recessive. So meiosis process, it will it will inherit one dominant from mother and one dominant from father. This is how natural selection takes place in the process of inheritance of gametes, which is known as meiosis, and this is how genetic recombination is done. So single member of each chromosome pair it receives is a matter of chance, and each reproductive cell then carries a random assortment of chromosomes and their respective genes. Then at fertilization, egg and sperm that unite are different from other eggs that are carried by mother and every other sperm carried by father. So this is how genes are recombined, which is known as genetic recombination. And this is done by shuffling of parents' gene. One cause of this shuffling is random segregation or sorting of chromosomes in meiosis. So I have already told you this random segregation or sorting of chromosomes. Chromosomes here it means in philosophical sense we can take it as genes. So genes are shuffled. It means taken apart. Well, I have given you example in which dominant and recessive they have been found out which are dominant, which are recessive, and out of this segregation the probability has been taken into account, and then the genes which were best for inheritance based on all the factors possible, because there are many factors, many processes that are played in background, and many factors that are considered by meiosis, which even the present molecular geneticists they do not know in its complete form. So we are still midway for midway in getting a whole idea of how genes and evolution and the inheritance of trait works. But we all we by now we know that genes they randomly segregate, and this random segregation, which is the genes, the whole lot of genes that are carried by parents in form of dominant and recessive, or you can say the assortment of genes in the form of those which are fit for inheritance and those which are not. So this segregation happens, and conceivably an individual could get any of the possible assortment of paternal and maternal chromosomes. So the genes or the chromosomes that are inherited by any offspring, it could be any of those. And which mathematics, which probability, or what factors are applied for inheritance, we are still in blind. Even the modern molecular genetics is still working on that. But as of now, we know that such process happened. Then another cause of shuffling of parental gene, it is crossing over, which is the exchange of sections of chromosomes between one chromosome and another. So this crossing over, it is also same as segregation and random assortment. So in which if there are traits, if there is a unit of DNA which is beneficial for offspring and which is not available in any of the parents, but the sections of DNA or the chain of DNA it exists. So such chromosomes they can be crossed over. So when I say that a particular trait it is functioned by, it is controlled by many genes or a long chain of DNA, or it may be there are different groups of chains of DNA which control a particular trait. So and we know that the chain of DNA it is also known as chromosome. So if by chance it happens that a particular chain or particular group of chain of DNA it is beneficial, but the derived matter it lacks that structure, it lacks that section of chromosome, then those section of chromosomes that can be crossed over from one gene to another. Because how does multiple groups of sections of DNA control and function a particular gene is still a work of modern molecular geneticists. But we know that The cellular function, they work in this way that if needed, any chromosome section, it can be crossed over from one gene to another. So this crossing over, it also compensates for the DNA or the chromosomes that are missing. Thus, after meiosis, the eggs and sperm do not receive just a random mixture of complete paternal and maternal chromosomes. Because of crossing over, they also receive chromosome in which some of section may have been replaced. So such replacement by crossing over, it will also happen. And it is all a matter of natural selection because the the offspring, he or she, is needed to derive the best of the genes, best of the traits, and best of the chromosomal structures. So the traits displayed by each organism are not simply the result of combination of dominant and recessive genes. Most traits are influenced by activity of many genes. Skin color, for example, is a result of several inherited characteristics. A brownish shade, it results from presence of pigment known as melanin. Then the degree of darkness in hue, degree of skin complexion, it depends largely on amount of melanin present and how it is distributed in the layers of skin. So this is also controlled and expressed by DNA itself. Then another factor that contribute to the color of human skin is blood that flow in the epidermis or the blood vessels that are in the outer skin of the layer. And human carry at least 5 different genes for manufacture of melanin. So here you can see melanin or the skin complexion itself. We may point out one hormone melanin or one pigment melanin, but the production of melanin itself or expression itself is both of them that depend on many genes of a group of genes. So the inheritance of gene in offspring, it is a complex process. In fact, almost all physical characteristics of humans are result of concerted action of many genes. Yes, so I may have explained many such characteristics or the whole idea in in this simple way, but it generally happens that, let's say, it is character A, but this character A, it may be controlled by a whole new group of genes which may be in this way. And all such genes, they by themselves might be working in the concerted in symbiosis with other genes. So a particular gene, it may be helpful for skin, but it may be helpful for teeth as well. So this concerted action, it its derivation, its inheritance, it is done by meiosis in a different way. And there are some traits which are sex linked. So the X chromosome, which together with presence or absence of Y chromosome determines sex. So it also carries gene for hemophilia or color blindness. So the expression of characteristic depends on the sex of the organism. Then the other source of variability, it is mutation. It is because mutation, it replenishes the supply of variability, which is constantly being reduced by selective elimination of less fit variants. So there is another strong idea that is written here, which is mutation. So we have seen how meiosis works. All in all, we get this idea that meiosis, it helps in inheritance of best fit genes. So if the best genes best genes are inherited. So what happens to those which are not favored? So those genes will be eliminated. So here an offspring, she derived two dominant variants of height from her parents, from mother and father, and both mother and father they carried both dominant and recessive alleles. So here both the dominants, the best genes, they have been inherited by meiosis, because this is how natural selection, it keeps selecting the best one, the best possible one. Then the here the less fit, the recessive one, they have been eliminated. They are no longer useful. So the offspring of this particular individual, they will not inherit those recessive genes. They are hereby eliminated from the lineage completely. So if natural selection keeps on going for a long time, so what happens that many traits that they are eliminated. So because of elimination, the supply of variability, it goes from diversity to uniformity. See here both are heterozygous. So it means both were diverse. They held the variations. Mother and father, both of them, they were gene diverse. But natural selection, it inherited the best one, best possible one. Now the diversity, it has gone. Uniformity, it has taken hold. So in order for natural selection to work, variations need to be there. And this may be a simple explanation, and the process itself, it is more complex than I have explained here. But yeah, on the basic level, it happens this way. And this may be that, like in numerical form, let's see, the composition of two individuals is this one. Now let's say any particular trait which needs to be copied only two numbers. So here the possibility itself, they increase manifold. But since an individual any individual, he or she, he possesses diversity of trait, and there are many traits or many genes that are in concerted, in constant and cumulative symbiotic actions. Genes work in conjunction and work with together. So here the traits, they are not to be seen in the form of alleles but random numbers. So here these are the numbers, and the possibility of deriving the best number, because the derivation here, I means inheritance. So inheritance, it is an exercise that brings uniformity. The best versions, the inheritance of best characteristic, this whole exercise, it is planked on the platform of uniformity. So if for many generations, you can say for thousands of generations, these process keeps going on. Let's see the example here. So here possibility are the best genes 1 1, then another possibility 4 4, then 6 6. So all the best numbers, they have been derived, and here we have run out of any possible number. So here again, the inheritance, it will integrate one dominant number, and then the out of all the other variables that are present here, best one will be integrated with it here. So now right now we have four possibility out of all these numbers. This is how the natural selection, this is how inheritance works. And natural selection, it works in opposite. Natural selection, it brings diversity, it brings variations, while inheritance, it brings uniformity. So if any individual, any population, any species has been acted on by inheritance in such a way that the final composition of the the present species, the present individual, it is more on the homogeneous side, more on the homogeneous or more on the uniform side. It means the variations, they have been eliminated. So if variations are eliminated, natural selection will gradually fade away. So to bring in more variations, to replenish the shortage of variability, mutations come into force. And then mutations, they bring much needed variability. So mutation is a change in DNA sequence. So, and such a change, it produces an altered gene. So majority of mutations are thought to occur because of occasional mismatching of chemical bases that make up the DNA. So here the thing that is being talked about is ATCG, the repetitive units. So if the structure of or the position of these bases are mismatched, it will produce mutation, because ultimately these units, they will combine up to form genes, which will combine to form chromosomes. Chromosomes will have forms of alleles, and alleles will be inherited. So the base one are these four bases, and if something, any structure, it is manipulated or tampered with in the basic unit itself, the changes will be manifold if the inheritance process, if it goes on. Then some mutations, they can have more drastic consequences, which depends on what portion of DNA this mutation is controlling. So as I have said, DNA is nothing but repetition of this unit, and any portion of DNA, any section or portion of DNA, it can form a gene, and there are many sections or many portions of DNA which do not manifest, which do not express in anything. They are just lies there. They are just there in the form of a unit, which sometimes the biologists call it junk DNA. So mutation itself depends on which portion of DNA is being controlled by, or which portion of DNA is mutated. The effect can be minimal if the product of the or the section, it hardly affect the organism. But if mutation happens at a place when DNA regulates the production of many protein, then this effect can be serious. So if this portion of the DNA, which is a gene for eye, and if it is mutated, then it will have lethal consequences. But if the same the same repetition of DNA, if it does not code for anything, if it is not for protein synthesis, if it is not for any particular trait, it is just a simple unit, and our DNA, it contains a lot of junk DNA. Junk, it means it doesn't mean they are not useful. It just means that we do not know what purpose do they serve. So that's why without any expression, without any code for particular activity, we call them junk DNA. So if a mutation happen at any such portion, then it will not be lethal. Then it is very difficult to estimate the proportions of mutations that are harmful, neutral, or beneficial. But some mutations have lethal consequences. Just say, let's see, galactosemia, which is caused by a recessive mutant gene and is result in mental retardedness and blindness. But it can be prevented by dietary restriction that begins at early age. So, but usually natural selection acts to retain only those mutations that aid in survival. Otherwise, all such mutations which are lethal, they will die off with the death of individual. Since if a particular mutation it acts on fertility, because most of the mutations which affects sex cells or gametes, they usually produce infertile individuals. Those infertile individuals, they will not produce any offspring. So such mutant genes, some such mutations, they will not transmit to further generation. And in that case, mutation itself will die off. Then let's see what Theodosius Dobzhansky had said, that consistently useful mutants are like needles in a haystack of harmful ones. But if needle is valuable, the task of finding it is facilitated by setting the haystack on fire and looking for needle among the ashes. So the role of fire is played by biological revolution of natural selection. So here in the cases that if a mutation is useful, but a useful mutation is also like a needle in the haystack, which means a rare one. Most of the mutations that result are either neutral or harmful. So to find the beneficial mutations, it's like looking for needle in a haystack. So if the task is cumbersome, its task is difficult, it can be facilitated if you set the whole haystack on fire, if you burn it, and which is like natural selection, because natural selection, the role of natural selection is similar to setting fire on useless things or traits which are not helpful, they are eliminated. The black moth that was spotted in Manchester in 1850 probably resulted from a mutation. So if the tree trunks had been light color, the moth and its offspring, it would have died out. They would not have any chance to survive because they will be visible against a light color background and will easily be preyed upon. So their survival, it was under threat, but industrialization increased tree trunks, they become darker and it acted as the maladaptive trait. It became adaptive, useful, and the population of black moth, it increased. So genetic recombination and mutations are source of new variations. But biologists have also identified two other processes known as genetic drift and gene flow. So genetic drift is the set of random processes that affect gene frequency in small and isolated populations. So uh, it also known as Wright effect after a geneticist Sewell Wright. So it is a process which may result in a neutral or nearly neutral gene becoming more of a less frequent by chance. and one variety of it is known as founder effect. It is when a small group of organisms recently derived from a large population migrate to a relatively isolated location. So if a particular gene is absent by chance in the migrant group, the descendant will also lack that gene. So here the case is, out of a large population, if a small population has migrated and stay isolated, in that case there will not be any genetic exchange between both the daughter and parent generation or the parent population. So if devoid of any genetic exchange or devoid of any fresh input of genes, that population, that isolated population, it will it will derive only the genes that are present in it. So if a particular gene is absent or present in it, the situation will remain same. And if all members of the migrant group, they are by chance carried a particular gene, then descendant would also share that gene. Since no intermixing is happening, no fresh inputs of genes is being advanced, then the genetic composition, it will remain the same. So the population that traveled over land when Bering Strait Bridge connected Asia to North America could not readily return. So this is about the Native American population that have higher proportion of individuals with type O blood than any other population. So this was also the first population of migrants that that connected uh, Asia to North America. So it might be that the North American population, the Native Americans, known as the American Indians, Amerindians, which have genetical and phenotypical composition different than the other white population. So it by biologists and geneticists, it is a hypothesis that these are the descendants of the first migrants who came from Asia to North America when Bering Strait or Bering Bridge, it provided them a ready passage, since it was a time when Earth was under ice. It was the ice age, interglacial age. So during that time, the frozen Bering Strait, it helped in their migration from Asia to North America. But during the interglacial, when the ice, when the atmosphere became warm and ice melted away, then they could not return. So in that case, the Native American population, they stayed there in American subcontinent, and because of genetic drift, their population, which predominantly have type O blood, without any intermixing with the American which lived there, and without any fresh supply of input from their parent population, they have inherited type O blood in a predominant way. Then isolation can also occur for social reasons as well, such as a religious sect of Anabaptists called Dunkers. They emigrated from Germany to United States in early 1700, and the fact that 15 original families kept to themselves, it explained why some of their gene frequency different from what both German and general US population. It is also a civil right effect in which the population, it kept to itself, its genetic composition, mm, it did not get intermixed with the native as well as their parent population. In that case, they came to derive uh, the composition, that they already possessed. And let's talk about gene flow. It is a process by which genes pass from one population to another to mating and reproduction. But unlike other processes of natural selection and genetic drift, it it tends to flow in the opposite direction. So a gene flow is a kind of a dilution in which the composition of the middle section, it is diluted other than the extreme sections. So here it decreases between populations. So two populations at opposite ends of a region, they may have different frequency of a particular gene, but the population that are located between them will have intermediate or diluted gene frequency. So it is this variation in frequency from one end of region to another, it is known as climb, which is also a reason of frequent intermixing with both the population. Since inheritance of gene, it is done by the the genetic composition already possessed by one individual. So in that case, the natural selection and the random segregation both will result in unique composition of individuals. So the intermixing and the intermediate gene frequency both are result of frequent intermixing with the intermediate population. Then let's talk about hybridization. So a species is a population that consists of organisms that are able to interbreed and produce fertile and viable offspring. Then individuals from one species do not successfully mate with individuals of or members of different species because of genetical and behavioral differences. So that's why human cannot produce offspring with horses and donkeys and elephants and other species. It is because of both genetic and behavior differences. And even if they can mate, such fertilization will not occur. And even if fertilization occurs, the embryo will not survive. And even if the offspring are born, they are usually infertile. So in here, the case is about two similar species. But the species, however similar they are distinct in way. It means there are some differences on the basic level. But recent studies, they have suggested that hybridization, the creation of a viable offspring from two different species, can be possible. So it may be another source of new variation in population, because hybridization, it will bring a whole new symmetry, a whole set of new genes in the otherwise uniform or homogeneous composition. So example here is the finches of this island, Galapagos Island, provide an example of hybridization in which female cactus finches, they died during a period of severe drought, which left abundance of males and high competition for mates. It led some female ground finches to mate with male cactus finches. Both were different species. And the results were hybrid offspring with unique characteristics. These hybrids were both male and female, but they went on to mate only with the cactus, the original parent population. So the end result was one-time influx of ground finch gene into cactus finch population, which added new variation. So otherwise, the process of hybridization, it was the same. Then let's talk about origin of species. One of the controversial aspects of Darwin's theory was suggestion that one species could over time evolve into another. So how does one species evolve into another? Speciation, the formation of new species, development of a new species, it may occur if one subgroup of a species find itself in a radically different environment, and in adapting to their separate environment, the two population may undergo enough genetic changes to prevent them from interbreeding. So, and numerous factors can prevent the exchange of genes. So, two species may live in the same area but breed at different time of year, or their behavior during breeding, the courtship ritual, they may be distinct. The difference in body structure of closely related form may itself bar or stop interbreeding. Geographical barriers as well may be common barriers to interbreeding. So, all in all, the basic idea is if one subgroup of a population of a species, if it is isolated from its parent population for a long time, then because of natural selection and differing environmental condition, both of species they may develop different characteristics. This will be a matter of time and natural selection both. But yes, if they are left to fend for themselves for a long time, so the difference of characteristic may lead to uh, the changes in the genetic structure, which itself will stop their inbreeding. If inbreeding or interbreeding, the genetic exchange, if it is stopped, then the population they are likely to proceed in their respective separate ways. So this is how new species can occur. Then the other question, do new species diverge quickly or slowly from their ancestor? Paleontologists they disagree about the pace of speciation. The traditional view was that evolution occurs very slowly over time, which is that the new species will emerge gradually with time, but others, which the supporters of punctuated equilibrium, they believe that species are very stable over a long period of time, but divergence, if happen, they are quick in nature. And let's talk about speciation versus creation. So some people who call themselves scientific creationists, they argue that although natural selection can produce variation within a species, it cannot produce new species because they support microevolution, which means variations, but they do not support creation of new species, which is macroevolution. And the idea behind that, they love for God, which is their emphasis on divine creation of all things. And an example by William Rice and George Salt, they demonstrated that if they sorted fruit fly by their environmental preferences, and then if the resultant groups are bred separately, they could produce flies which are incapable of interbreeding, which means separate species as in as little as 35 generation. Then the fossil records, it also contains numerous examples of species, speciations, and development of new species. Here, another example by fossil record is about evolution of bird from terrestrial creatures. So the complexity of life, these creationists, they argue that must stem from intelligent design, which means they are hailing God here. They want to see the footprints of God, and even if they could not see, they want to put the derive this idea in theology. So according to them, the creation of the creation of new species, new individual, uh, it should be a work of divine creation, of divine entity here, which means God. And The intelligent design is at the core of political movement. The problem, however, is that intelligent design cannot explain speciation in a scientific way. So it's a personal view, it's a theological view, which, if integrated into science, must stand the scrutiny on the anvil of practical, on the anvil of rationality. It must be tested and seen. And if it is just a matter of faith, then faith is a different matter altogether, and in that case, it should be separated from scientific approach. And they argue that if natural selection may not account for all cases of speciation, then only alternative is divine intervention. So here, their argument is that since the present status of science, it cannot provide all the answers of science, then the actuator here, or the hand that set off all the unanswerable question, was something divine, which is a flawed analogy, flawed reasoning, because just because we do not understand something today does not mean we should toss out all we should know, or we should put all on the shoulders of a divine creation. The scientific approach, it is to continue examining the unexplained phenomena to learn more about them. So if we don't know anything about something, it's a matter of time, it's just a matter of time before we will know about it in much greater detail, but still we will have something left which will be catered to, which will be addressed, or which will be looked into by the future generation. And then the key facts which are appealed by intelligent design creationists are the biblical one. So then, science cannot rule out such possibility because they cannot be observed, measured, or experimentally tested. Then science and religion should be regarded as different way of understanding the world. The former, it relies on evidence, while religion, it relies on faith. So faith and experimentation both should be stayed in separate affairs. Then let's talk about natural selection of behavioral traits. So we know that mm, physical traits, they can change in a population. Physical traits, we have evidence of natural selection acting on it. But what about the behavioral characteristic of population? So can they be inherited? So the approach called sociobiology, behavioral ecology, evolutionary psychology, and dual inheritance theory, they apply such evolutionary principle on the behavior of animals and humans to see if natural selection also act on behavioral traits. So here, behavioral ecology, it looks at how all kinds of behavior that are related to environment. Sociobiology, it looks at social organization and social behavior. Evolutionary psychology, it looks at evolution have produced lasting variation in the way humans behave, interact, and perceive the world. And dual inheritance theory, it looks at how beneficial cultural trait might be selected for and transmitted. An example here, consider the lion. So it is also a member of feline or cat family. So all the members of cat family are normally solitary creatures, which means they like to live alone. But lions, they live in a group called pride. So here, this behavioral phenomena, the behavioral change, it might have happened because of evolution, because group hunting is most successful way to catch large mammal and open terrain. Then the social behavior of lions may have evolved primarily because it provided selective advantage in lions' open country environment. So one example here, it is about the geography or how nature acts on behavior of people or behavior of organism. So cat family, it stays alone. Most of these species, most of the individuals, they stay alone, but lions live in group. So one um, postulate, one reason that has been provided here is that since lion, they live in open country environment, and in that case, social hunting or group hunting, it comes it becomes an advantageous activity. So it might have that nature has integrated or inherited such behavior, such social behavior in lions. Then here in the moth example, the color of moth is part of its phenotype, which which can be subjected to natural selection, because we integrate, we can understand this, that anything that is visible, anything that is expressible, such as the way we look, the way uh, the positions of eye sockets and head and skull and bones and skeleton and all such organs which are visible, they can be acted against. But behavior is not any any such thing which can be seen easily. Behavior, it cannot be given a concrete form. So how can behavior be selected? But then behavior is also an express characteristic. If hunting in group, behavioral trait gets you more food, then individual who hunt in group will do better. So it may be that such characteristic, it has been selected against by nature. Then most biologists, they understand that express or observable characteristics are the results of gene, environment, and life history all interacting together. The sociobiological approach, it has aroused considerable controversy in anthropology, because probably because apparent emphasis on genes rather than experience and learning. And anthropologists have argued that the custom of a society may be more or less adaptive, because cultural behavior also have reproductive consequences, because a cultural mechanism, all in all, they help in survival, they help in coping up with the atmospheric conditions. So those cultural behavior, they will have a say in reproductive success. Then the question is, how do cultural evolution and biological evolution, both of them, they can compare to each other? To answer this question, one must remember that operation of natural selection required three conditions, and the three conditions are variation, heritability, and a mechanism that duplicate traits in offspring, and differential reproduction of heritable differences. So the three basic conditions: variations, then heritability, and third one, differential reproduction. How does the three conditions can apply to cultural behavior? So in biological evolution, variability, it comes from genetic recombination and mutation. But in cultural evolution, variability, it comes from recombination of learned behavior and from invention. Then cultures are not close or reproductively isolated. A species cannot borrow genetic trait from another species, but a culture can borrow new things and behavior from other culture. So the difference here about both um, differences here between cultural evolution to that of biological evolution, and all the three conditions are not being met. Variations can come, but the borrowing element, it is absent in biological side, whereas it is present in cultural side. And then both heritability and differential reproduction, both are clearly absent from cultural agenda. Then children who learn by imitation or by parental instruction. So children and adults may also copy adaptive traits they see in people outside the family. So here um, there is another glaring difference between culture and biology, since children who copy their parents, the genetic code or the genes, they cannot be copied by people who are outside the family. But cultural traits, they can be copied, they can be borrowed. Then comes the requirement of differential reproduction. Uh, and it does not matter whether the trait in question is generic or learned or both. And as we emphasize, behavioral incompetence lead to extinction as surely as does morphological disproportion or deficiency in any vital organ. So here, when we talk about differential reproduction, here um, we know that biological incompetence, which means if you do not have the appropriate morphology, if genes are defected, if you possess deficiency of any vital organ, then such incompetence or such shortages, they cannot be compensated, they cannot be refilled, and presence of such will certainly lead to extinction of an individual. And the same thing applies to cultural elements of behavior as well. If your behavior is incompetent, if you behave in a such way that it is not culturally adapted, if the behavior, all the cultural traits that you hold in your arsenal, all of them do not fit against the nature or the environment in which you live in, then certainly they will act against you. Then let's see the dual inheritance theory. They distinguish cultural transmission by learning and imitation from genetic transmission. So here, William Durham also deals separately with cultural transmission using the term meme, which is analogous to gene for unit of cultural transmission. So here, the basic idea for them is that human being, they inherit both genes and cultural elements, and both of these, they are transmitted down the generation. Genes are transmitted, genes which are basic unit of biological traits, they are transmitted along with memes, which forms the unit of cultural transmission. Then some of the most important biological features of humans, such as our relatively large brain, that may have been favored by natural selection because our ancestors made tools a cultural trait. Conversely, cultural trait of informal and formal education may have been favored by natural selection because humans have long period of immaturity. So here, both biological and cultural trait, or you can say whole dilemma of transmission of both biological and cultural trait, it has been set to rest in a way that both of the traits, they are similar, and both of them, they help in survival of an individual and species. So both of them, they compensate each other. And let's see the review. First, evolution of evolution. So ideas about evolution that long time to take hold because they contradicted biblical thinking about divine creation. Then in geology, the concept of uniformitarianism, it suggested that Earth is constantly subject to shaping and reshaping by cultural forces. Then Charles Darwin and Alfred Wallace, they proposed the mechanism of natural selection for evolution of species. And let's see the summary of principle of natural selection. The process of natural selection, it increases proportion of individual, increases the survivability of individuals within a species because of advantageous traits, and those advantageous or adaptive traits that result in great reproductive success within a particular environment. And maladaptive traits will eventually decline in frequency. Then natural selection does not account for all variations in the frequency of trait. Two types of natural selection, three types of directional, normalizing, and balancing selection. Then heredity. Gregor Mendel's research on pea plants helped scientists to understand biological mechanism by which traits may be passed from one generation to the next. And the basic unit of heredity are genes. Genes occurs in pairs in the form of alleles, and genes are inherited through sperm and egg cell, which are created in the process of meiosis. Genes, which are made up of DNA, provide instruction for cell to make proteins. Proteins are long chains of amino acids that make the structure of organism and drive life processes. The segment of DNA are transferred from cell nucleus by mRNA. This mRNA is then read by ribosome in the cell to construct proteins. And let's see the sources of variability. Natural selection, it depends on genetic variation within a population. And new variation can occur through genetic recombination, mutation, hybridization, genetic drift, and gene flow. So the process of genetic drift and gene flow, which are genetic drift, is described as the set of processes that affect gene frequency in small and relatively isolated population. That such migrant population, they are likely to derive the, the genes which are present in them. Since intermixing or interbreeding is not there, so if if some gene is present in them or absent in them, their offspring or the whole population will also come up with the same presence or absence. Then gene flow is the process whereby genes pass from one population to another through mating and reproduction, in which the population on the middle or the population in the intermediate edge will have different set of genes, or the genes in the intermediate range. Then origin of species, which is speciation, which means development of a new species, may occur if one subgroup it becomes separated from other subgroups, and while adapting to different environments, they may undergo enough genetic changes to prevent interbreeding. And the absence of interbreeding itself suggests that two different species have arrived. Then natural selection and behavioral traits. The natural selection can also operate on behavioral characteristic of population, and approaches such as sociobiology and behavior ecology they involve the application of evolutionary principle to the behavior of animals and humans. And much controversy surrounds the degree to which natural selection can be applied to human cultural behavior. So this is it. Thank you very much. Kindly subscribe the channel.